So with that, we're going to move on to our next presenter, who is Andrew Farmer, is the Vice President of Research at National Science for Genome Resources in New Mexico, in New Mexico. And he's going to talk to us about how to integrate multimodal data sets. So Andrew, the floor is yours.
Okay, thank you, Kevin. Screen. Okay, so thanks everyone for letting me spend some time with you. As I said when I accepted the invitation to make this talk, I really feel like I'm far from an expert in this subject, but I have been lucky enough to have the opportunity to work with some both RNA-seq data, single-cell plant RNA-seq data, and ATAC-seq data. And so it's definitely a work in progress as far as what we're doing. And I'm happy to just share some of the exploratory things that we've been doing in this space with you. And so what I'm really going to talk about is sort of the difference between unpaired and paired multimodal measurements. Okay, so what does that mean? So when you're talking about multimodal data, you have a variety of ways that you could have those modalities come together. And the two main ways I think that we're going to be thinking about are so-called paired integration, where you actually have measurements of two or more modalities on the same set of biological entities, whether they be cells or nuclei, or unpaired integration, where you actually you have two different modalities like RNA-seq and ATAC-seq, but you're doing them on a separate set of cells and then making some assumption that allows you to bridge the information between them. I think other speakers have mentioned these best practices consortia that are out there. There's a nice paper out concerning best practices for multimodal data, and I'd encourage you to look at that. But this other paper came out recently, very recently. I just happened upon it when I was thinking about this talk. And it was specifically focused on the questions between how well does paired multimodal data do relative to unpaired in the context of RNA-seq and ATAC-seq data. And interestingly, one of their conclusions was that one thing that's really critical in terms of getting the most value you can for certain tasks, at least like cell type annotation in the context of true paired multimodal data, is really having as many of the cells or nuclei as you can. And when you're talking about single ATAC-seq data, you're really talking about single nucleus data. And this question about how do you tell good quality nuclei from bad quality nuclei, and how do you increase the numbers that you're working with for some of these things where you're really dealing with sparse data, seems to be a continuing theme from a lot of the speakers. So I'm happy to be able to maybe add a few perspectives to that. Okay, so I thought maybe I should give you a quick overview of what ATAC-seq is all about. This is the original paper that presented the method. And essentially what we're talking about is the fact that in the cell, the DNA is largely wrapped around these histones. And in order for the transcriptional machinery to really be able to cause genes to be expressed, the transcription factors need to have access to the underlying DNA. And so there are many different techniques that allow you to be able to assess this notion of which regions of your DNA are accessible or not to the protein machinery. But AttackSeq has become pretty popular because of some really nice characteristics it has just as a lab protocol in terms of really being quite good in terms of having low starting material and also being tractable in terms of the protocols. So it's really become one of the leading methods out there for looking at open chromatin. All right, and so a couple of years ago, most of my work, if not all of my work in this space, is done in collaboration with Mark LeBeau's lab. And Mark and I have worked together on soybean RNA-seq for many years, but he approached me and said, I'm interested in looking at single nucleus data. And one of the reasons that we started thinking about nuclei rather than protoplasts, like everybody else, we sort of started with Arabidopsis roots, but we were thinking about nuclei as a nice way, avoiding the issues with protoplasting that other speakers have mentioned, specifically with an eye to sort of doing it across multiple species and thinking that would be, nuclear isolation would be much easier. And so this first paper that we came out with focused on nuclei, and a lot of it had to do with just establishing that nuclei could give you comparable results to the single cell protoplast data. But in addition, since AttackSeq, single nucleus AttackSeq is done with nuclei, it gave us a good opportunity to create an unpaired set of data with RNA-seq on one set of nuclei and AttackSeq on another set of nuclei. And so this is just a kind of an overview of the protocol that we used. A lot of it was really fairly standard in terms of using the toolkits that were available at the time, Seurat for the single nucleus RNA-seq, and then the kind of companion package called scSeq, which was dealing with the AttackSeq side of things. And AttackSeq data has some special properties. One of the previous speakers kind of alluded to the fact that it's really highly sparse, and almost you can in many ways think of it as a binary thing where you're just sort of thinking, is my chromatin at this peak open or closed? There is some debate, I think, in the field about whether treating as a binary state or a quantitative measure is better, but a lot of the methodologies, will handle it in a similar way to things like the Google indexing system, where you're using a technique called term frequency inverse document frequency normalization, and then applying latent semantic indexing as kind of an alternative to the way that PCA is used as the basis for producing the initial reduced dimensionality on the RNA-seq side. In any case, when you're doing the integration of the two, because you're not making the measurements on the same things, you do have to make this assumption that open chromatin is going to be reflective of gene activity. And so there's a point in this protocol where basically you create from the AttackSeq data something that translates the feature space from peaks, which are the kind of unit of features in AttackSeq data, to the genes that they're nearby, and you sort of convert it into a gene activity matrix, and then you perform an integration that sort of thinks about the whole set of cells in terms of gene space rather than two separate sets of features. Okay, and this we were able to kind of do some comparisons that showed that in fact, you could look at the AttackSeq data independently of the RNA-seq data and get a fairly similar, at a high level, structuring of a UMAP and similar number of clusters. And when you make this assumption about the the correspondence, you can then sort of assign cluster identities to your nuclei from the ATAC-seq based on your RNA-seq data, and then you can look at correlation structures. And we found some interesting things. We were able to sort of show that our ATAC-seq data largely was similar to the overall picture that bulk RNA-seq or ATAC-seq, sorry, presented. But in addition, you could tease apart, as you would expect, cluster-specific peaks that seem to be related to gene-specific activities in some of these clusters. And in addition, one thing I think the first speaker kind of mentioned the fact, when you're dealing with nuclei, there's a sense in which you are losing information because you're excluding a lot of the data that's in the cytoplasm, but you're also focusing on the data that's kind of closer to the action in terms of the active gene expression. And so one thing that seemed to be reflective of that was the fact that when we started looking at correlation between the RNA-seq data and the ATAC-seq data, it was a better correlation that we had with the single nucleus data than with the single cell data that we had integrated into the same clusters. Okay, now a lot of what I'm going to talk about kind of is going to be along the theme that other speakers have talked about in terms of, well, how do you actually decide what are good quality entities to include in your analysis? And I guess we all sort of start with Cell Ranger or one of its variants like Cell Ranger ARC, which is what's used in multimodal data. And then we find that it does things that we're not all that happy with, and we start looking for alternatives to thinking about, well, what should I include or what shouldn't I include? And one of the previous speakers mentioned this GRAS transcriptome, where they also looked at single cell versus single nucleus. And one of the conclusions they came to was that you basically need to double the number of nuclei because of the sparsity of the count data that you get from nuclei in order to recapitulate similar clustering. And that's just provided as a kind of a general rule of thumb, somewhat similar to what we had seen. But there are sort of downstream analyses that you might want to do that really, in terms of the statistics that you need, having more counts associated with clusters is a good thing. So one thing that we're interested in, because of some of the species that we're interested in being polyploids, is looking at things like the ways in which gene duplicates have diverged in their expression over time or evolutionary time. And so some of the statistics behind that, it's going to be very underpowered if you don't have sufficient counts. And so the more things you can get from your experiment assigned to clusters to create these kind of pseudobulks to look at counts for, the better, as long as those are actually good cells and properly assigned. And then one of the issues that you have when you're dealing with the unpaired data is that you can kind of cluster things separately and use these integration techniques to project your assignments from one space into the other, but you don't necessarily get concordant results when you're doing that. And so what I'm showing here is sort of the UMAP produced from the ATAC-seq data alone on these nuclei and different clusterings, one of which is done in the RNA-seq space. And that's kind of presented in terms of the numbers that are given down here, and then the clusters that you get when you're thinking about it just using the ATAC-seq data. Those are represented by the colors. And so, there are some clusters that are really nicely concordant between the two, like these ones that I've put stars on, where you have just a nice simple one-to-one correspondence when you look at the cells and what they're being assigned to on the one hand versus the other hand. And then you have the two other possible situations where you have sort of splits from the one modality to the other or merges. And this way of looking at it, I think Mark probably spent some time doing this all manually, but you can basically create these so-called confusion matrices and get a more quantitative view of what's going on between the two modalities. And what we're seeing here is a sort of heat map representation where the RNA-seq assignments are on the x-axis, the attack-seq cluster assignments are on the y-axis. And you can either just look at it in an unscaled way where the total counts are showing by their colors kind of the overall structure, or you can scale it by row and see things like the attack-seq data kind of being split out into clusters that correspond to only one RNA-seq cluster, or the opposite way when you're looking at it scaled in column space, in this case, where I've got the RNA-seq data on the x-axis. All right, and so this kind of gives the basic idea of, well, why would you want to start thinking about getting paired measurements? All right, and so in fact, there are kits that allow you to do paired measurements, and I think Sandra Tavillier from Mark's lab presented on these protocols in the previous workshop, but essentially the idea is that you're doing the two RNA-seq and attack-seq protocols on the same nuclei and creating then the two separate libraries that you sequence in different ways. And as I understand it from Sandra, there's kind of a balancing act that you need to do in terms of the library prep to get, you need to permeabilize the nuclear membrane so that the transposase is actually able to get in and do its thing. But if you permeabilize too much, it has kind of bad implications for leakage of RNA-seq. And so this leads to a situation where you've got these two modalities, and you may have a nice representation of the one, and not so great representation of the other. And it really kind of complicates the question of how do you decide which of the nuclei you want to include in your analysis. And furthermore, in some contexts, well, you can see that I've got kind of a really unrealistically high number of nuclei that Cell Ranger called 20,000, which is like the highest number it will call. And this actually happened kind of after I added organelle representation to my reference. And so there are a lot of really kind of weird things that can happen in terms of the cell calling, at least just using the standard methods that are provided by the 10x tools. And you'll also see here, we're dealing now with soybean nodule data, and I have a really low number of reads that are mapping to the reference. And that's probably not all that surprising if you know anything about nodules, because they're really packed with bacterial DNA. So the symbiosome is sort of within the infected cells within the nodule. You can kind of see there's really not a lot of room left in the cytoplasm once these things have matured, and they're filled with these symbiosome structures that the bacteria create. And this is shown in some nice spatial data from Sutton Tennant in Mark's lab, where the yellow gene here is actually a bacterial gene, and you can see that it's pretty much filling up the whole infected cell, except for in the nucleus, where I've got a soybean marker gene to just kind of show that those are really nuclei expressing marker gene for the infected cells. So the presence of all this bacterial DNA, it's kind of interesting that the cell walls of the bacteria in this context are actually permeable enough apparently, that you lose a lot of data to the to the bacterial DNA. And this has been seen by others who've looked at or tried to look at ATAC-seq data in the context of nodule. And so in the case of Arabidopsis, we had a lot of bulk data that we could compare to decide, are the peaks that we're calling out of this sort of looking similar to what we see in the bulk data that's been produced, whereas here in the context of the soybean nodule, it's a little bit more difficult to really decide, whether we're seeing things that are accurate or not, because there really isn't a standard for accuracy. Although we do see some regions that seem to be fairly consistent with some of the tissues that were produced within this publication, and others where there's not so much consistency, and that may just be due to the fact that it's a very different tissue. All right, and so again, Cell Ranger, has its own notions about how to approach this question of what things to include in analysis. When you're dealing with this 10x multi-ohm data, you can either run it as a multi-ohm, which I've done here in the center, and then I've taken the same sample and run it in expression-only mode or attack-only mode, and you can see that the number of cells being called is very different in all cases. And this is because the interpretation of what's going on from the counts in these droplets is very different, based on these knee plots that the previous speaker was describing in detail. The calling that they do in the combined context is a little bit different. It's kind of looking at both modalities together and trying to use them in a joint way. But it's a little hard to really know, what, if there is this trade-off between, permeabilization, possibly leading to RNA-seq loss, how should you approach this? And one thing that we found is actually a pretty good standard is just kind of looking at the clustering that you get out of the different modalities. And if one is sort of more highly structured than the other, you might decide that the one modality is preferred in that particular library. We do see, you can kind of look at the molecular counts independently and can see some kind of interesting things. I think, largely, generally speaking, you see similar trends in terms of the different clusters and the molecular counts associated with them. This cluster 5 is kind of interesting in this case because it's clearly showing a kind of a different behavior, and I think that probably reflects the fact that there's a lot of different endopolyploidy levels within the cells in that particular cell type. But you do see also some cases where we've got a cluster where the behavior of the ATAC-seq doesn't really seem to be matching all that well in terms of just overall counts with the corresponding level of RNA that we're getting. Okay, so in terms of QC for ATAC-seq, there are some different types of measurements that people think about. So in addition to just kind of overall counts of the molecules that you get, you would like to see that a lot of them are associated with the transcriptional start sites. And I'm just showing here that there does seem to be a fair amount of difference in terms of the actual values of transcriptional start site enrichment between different species. This is probably something that's been observed in bulk ATAC-seq contexts. I guess it was a little surprising to me to see that soybean would have kind of higher values in general than Arabidopsis, but that's at least what our data seems to be telling us. And we can see that, in terms of the different replicates that we have for soybean, some of them are fairly consistent, and then this one seems to have a lot more of the population at a lower level of enrichment. And then again, you can also look at these sorts of things from a per cluster basis, and there's a little bit of variation, but it seems fairly consistent among the different clusters. Another thing that's kind of interesting to look at when you're thinking about them as just kind of separate modalities, even in paired context, is just kind of how the different modalities behave on their own. And so, you can just basically put them together without doing any special batch effect correction and see how they map together into the UMAP space. These are the merged versions. Oops. And the integrated version is applying these kind of batch correction effects. And it's kind of interesting to see that, at least in this case, there seems to be quite a bit more batch effect in the gene expression data than there is in the ATAC-seq data. And some previous speakers have also mentioned this issue of single nuclei data being kind of prone to ambient RNA. And we'll look at that in a little bit more detail soon. So one of the techniques that's been developed for dealing with paired data is this so-called weighted nearest neighbors approach that was introduced in Seurat version 4. And the basic idea there is that you take each cell or nucleus in this case, and you look at its neighbors in the nearest neighbor space of the RNA-seq and the nearest neighbor space of the other modality, and you just look to see how predictive of the profile of the cell that you're looking at are the nearest neighbors to it. And based on that level of prediction, or however representative the nearest neighbors are, that allows you to assign kind of a weighting to the two modalities in terms of how informative is each one for the particular cell in question. And in some modalities, like for example, with scSeq, where you're looking at proteins versus expression, it's been shown that there are some cell types where they're not really all that well differentiated with respect to, say, their expression content, but there are surface protein markers that distinguish them pretty well. I think in general, it's not that much the case that ATAC-seq data and RNA-seq data is expected to be all that differently informative in different cell types, although there could be interesting cases where that is true. But what we found is that it actually provides a kind of a nice QC as to how good a quality the different modalities are in your particular library. And so we can see we're just looking at the gene expression being more highly weighted, towards the one, whereas if things were around 0.5 in this case, it would mean that they sort of had equal weights given to the gene expression versus the ATAC-seq, whereas things higher to 1 in this case are looking better from the RNA-seq perspective. And so we can see that one of the libraries seems to be fairly poorly weighted towards the ATAC-seq, whereas others are largely more balanced towards that kind of median at around 0.5, although there are certainly cells or nuclei in the other cases where they are looking better in terms of one modality or the other. Okay, so we started having questions about the effects of doing filtering. So, as I said, I had kind of heard about the fact that nuclear data tends to be prone to this ambient RNA phenomenon, which is kind of, when you think about it, it's expected when you're lysing the cells to get at the nuclei, you would have a lot of extra stuff. And so we started sort of thinking about, well, what is the effect of this ambient RNA? How can we correct for it? And what's the overall effect of other filtering on the structure of UMAPs and clustering? So here's an example of a data set, where the mixed cells of the first speaker are sort of very prominent here. Then when you do the ambient filtering and some noise reduction of the input cells, you get much more nicely separated clusters. Now, whether or not that's important for the particular question that they're looking at, I think is another topic. But anyway,
Hey, Andrew. Just a quick. Maybe you can wrap it up, maybe like 30 seconds, so that we can get to
oh
questions.
boy, okay, sure, all right.
No, you're good.
Okay, well, so this is basically the cell bender tool that does, it has a very complicated probabilistic model that's modeling a number of different sources of noise, as well as the question of whether or not cells are actually cells or empty droplets or debris containing droplets. And they've shown that it works quite well in terms of nuclear data, which is one of their focuses, and also can be used in terms of different modalities. So we've started applying it to some of our data, and found that it does a nice job of identifying things that you might expect to be ambient in this context, like the organelle data. We're trying to then incorporate it into views like the C loop with this new package called Loop R that's come out of 10x. And there are other tools. I'm going to just breeze through this. They do claim that it could be useful for AttackSeq, particularly in terms of certain phenomena like barcode swapping. That's a consequence of mixed PCR in the droplet space. But we haven't really explored that all that much at this point. But when you apply it to clustering, you can actually see you do get cells that weren't present in the initial data sets, sort of based on cell ranger output, clustering fairly nicely and included in there. Whereas you also do get large clusters of things that are highly enriched for organelles. But finally, you can just start looking at the presence of these things in terms of the different clusters. Certainly, there's a lot of stuff that's highly enriched in these kind of spurious clusters, I think, but also other clusters that seem to get enriched for some interesting content. And then you can also start looking at the different modalities against the weighted nearest neighbor space and see how well they do. And finally, there are a lot of other things you can do with the AttackSeq data that maybe I should have spent more time talking about, but things to explore in particular, things like looking for transcription factor binding through footprints in the AttackSeq data. One thing that I've found is that some of these nice toolkits, it's actually pretty hard to get some of the plant genomes to work well with them. So don't underestimate the amount of time that you might spend trying to get your favorite genome to work with some of these tools. And finally, the basic idea eventually is to take the knowledge gained from the specifics of the AttackSeq and combine it with regulatory network analyses, which other folks from Mark's lab are doing. And we're also interested in sort of how do you incorporate this data into public resources like the legume information system that I work for. So various tools that we have that we're trying to kind of put this data into. And with that, I'll end, and sorry for going a little bit longer than I should have.
Oh, awesome. Thank you, Andrew. Just as a quick reminder, feel free to put any questions you have for Andrew in the Q&A. So one we have here is, so when the clustering leads to say one ATAC cluster corresponding to two RNA clusters, if you look more closely in subclusters, the ATAC clusters, do you know if you get concordance or the same cells with those two RNA clusters?
Yeah, I think you don't really know that when you're dealing with the unpaired data. I guess what you could do is to sort of look at the two clusters and the impact of using the one versus the other on the peaks that you get out of those two. But I think it's only really when you have the paired data that you're really able to assess the extent to which the different clustering schemes based on the separate modalities, concur with the combined sort of analysis.
Gotcha. We have another question that asks, can you provide more details about the cell veteran plus ATAC-seq?
A very little bit of detail. So if you go to their GitHub, you'll see that there are some issues open about whether or not it's appropriate. And basically, I think the overall idea is that you probably aren't going to get a lot of ambient noise from ATAC-seq, but this other phenomenon that they address in their model, which has to do with barcode swapping among the cells, they do think it's appropriate for that based on some of the other phenomena that they've looked at. And so they do allow you to sort of apply the model with different parameterizations that allow you to sort of turn off the ambient and only look at the swapping. And so that's the direction I'm going to be starting to look at it in.
Awesome. Cool. Well, in the interest of time, I think we're going to move on to our next speaker. So Andrew, thank you again.